Happy Friday. Very good, very good. I have you trained. Happy weekend. No exam this coming week. Finals beckon. Okay, so we're moving along talking about transcription. Last time I got started talking a little bit about eukaryotic transcription. I'm going to say a lot more about that today. And a lot more about modification of eukaryotic RNAs. And eukaryotes are interesting in comparison to prokaryotes in that respect, particularly for messenger RNAs. We see a lot of modification to messenger RNAs that happen in eukaryotes that don't happen in prokaryotes. Before I get started with that, though, I want to remind you, I said last time that eukaryotes also have, like the prokaryotes do, a TA-TA-rich sequence region near the start site of transcription. As I said, in eukaryotes, it's a little bit more variable in position relative to the start site, typically within about 100 base pairs or so of the start, and not exactly 100. It's usually in between 30 and 100 bases away. It's not always there. Not all genes have that, but genes that are made at reasonably high levels will almost always have a TATA box of some sort. There are other sequence elements that are important in eukaryotic transcription. A couple of them are here, cat boxes, as you see here, GC boxes, as you see here. Again, I'm not expecting you to memorize the sequences here, but these are conserved elements that are found in many promoters of eukaryotic genes. Okay? Now, eukaryotes are quite a bit more variable than prokaryotes are in terms of the variability that we see of promoters. Very, very. You may find quite a few genes in eukaryotes, for example, that don't have a cat box. You may find some that don't have a TATA box. You may find some that don't have the GC box here. You also see some variability in the positioning of these relative to the start site. So it is a more confusing picture in eukaryotes. And the most important thing that we can be thinking about is just what are the sort of sum of all the elements that are there. Another thing that pops up in eukaryotes is something called enhancer sequences. I'm going to jump ahead to that at the moment and say a word about that before I say something about transcription initiation. Enhancer sequences, or enhancer sequence elements, which would be the same thing, are sequences that affect transcription in eukaryotes, but they have a very, very different character than any kind of sequences we see in prokaryotes. Okay? Your book lists some examples of what they call prokaryotic enhancers, but really that's not a very accurate description. And you'll see why when I describe this in a second. Okay? So enhancer sequence elements essentially are only found in eukaryotes. And they are typically short sequences that we see that appear before a variety of genes. But the big differences that we see are, A, these elements can be located in very wide-ranging positions relative to the start. It's not uncommon to find enhancer sequence elements for a gene a few thousand base pairs ahead of the start site, minus 3,000, for example. Okay? It's also possible, in some cases, to find enhancer sequence elements that are downstream, that is in the plus sequence region of the start site. That's something we haven't seen before. So there are some that may be in coding regions or in other regions ahead of where that start site is. Now, enhancer sequence elements also, as I said, can be very variable in their relative position. And one of the reasons we don't really see examples of this in prokaryotes is because prokaryotes, the genes are very tightly packed together. There's usually not a lot of big gaps of thousands of base pairs of sequence where there's not another gene. In eukaryotes, because the chromosomes are much bigger, the genes tend to be much more spread out, and there are a lot more gaps between them. So having a sequence control element a few thousand base pairs away is not a big deal. Okay. Now, I'm not going to say exactly how these work here. I will say something more about them when I talk later about gene expression. But I do want to point out a very important thing about them. The sequence elements appear to play important roles in conferring tissue specificity to genes. Tissue specificity, all right? So if, for example, I have a gene that needs to be expressed in muscle cells, then it very likely will have a sequence element that is bound by a protein that's found in muscle cells. Okay? And that protein that's binding that sequence element in muscle cells will help to activate its transcription so that that gene is made in muscle cells. Right? That same gene, of course, is present in all the cells of our body. In bone cells, if there's not a sequence element for bone cells, that same gene may not get made. If it needs to be expressed in bone cells, then there may be, in addition, another sequence element that's upstream that would have a bone sequence protein binding site. All right? So using these sequence elements, cells can actually specify where and when those individual genes are transcribed. That's a really important thing because the needs of bone cells for proteins are different than the needs of muscle cells or skin cells, okay, or intestinal cells, you name it. So having these different elements that are there and different proteins that can bind them allows the cell to give some specificity to which proteins are being made in which cell and at what time. Well, as we talk about transcription, I want to just say a brief word about initiation. And what I'm going to show you is one way in which the transcription of eukaryotic messenger RNAs can occur. And it's not the only way, because you'll see that this way I'm going to describe to you shows the use of a TATA-Binding protein, and not all genes, of course, have a TATA site. If you recall from what I said last time, RNA Polymerase II is the polymerase that's involved in transcribing messenger RNA. And we see something different than we saw in prokaryotes. In prokaryotes, we basically had an RNA polymerase, and if it had a sigma factor on it, that sigma factor helped it to recognize and bind to a promoter sequence. In the case of RNA polymerase, we don't have such a setup. In fact, what usually happens in eukaryotic cells is that there are a host of other proteins that are involved in binding, recognizing and binding to the promoter region, and then only after a complex of these has been assembled does the RNA polymerase come in and do its thing. So that's depicted here on this screen. As you can see, there are a sort of an orchestration of the activation of transcription going on in this gene. Well, the first thing we see is that there's this factor called TBP that stands for TATA-Binding Protein. And TATA, of course, is that AT or TA-rich sequence that is ahead of the start site of transcription in eukaryotes. And TATA-Binding Protein is one of the proteins in a larger transcription factor called TF2D. That's a Roman numeral 2, TF2D. TF stands for transcription factor 2. Of course, we're referring to RNA polymerase II. And D is just the name of the protein. Okay? So TF2D contains, it has proteins, and one of those is the TATA-Binding protein. TF2D is the first transcription factor that's binding to this promoter. Now, when I say transcription factor, I want to remind you that a transcription factor is a protein that recognizes and binds to a specific sequence in DNA. Recognizes and binds to a specific sequence in DNA. That transcription factor may activate transcription. That transcription factor may inhibit transcription. So for our purposes, all we care about is that it binds a specific sequence and in some way it affects transcription. Well, in this case, it's activating it because we see TF2D binding. And then we see this TF2A coming in and binding. We see TF2B coming in and binding. We see the RNA polymerase come in at that point, followed by TF2F, TF2E, TF2H. We're not going to worry about the individual functions of these proteins, but I just show you this to plant, hopefully in your heads, the orchestration the fact that there are other proteins that are binding before RNA polymerase comes in and does its business. And only after all of this complex has been assembled does transcription actually start. Okay? Now, one of the things that you'll notice in the process of this is that there is something called the CTD phosphorylated. The CTD is this thing right here, and CTD stands for the carboxy terminal domain. Excuse me. That's the carboxyl end of the RNA polymerase. In order for transcription to proceed, that phosphorylation has to happen. So the RNA polymerase must be phosphorylated in order for the RNA polymerase to move forwards, and it moves forwards as you see here. Excuse me. The TATA-Binding Protein is shown here, complexed with DNA. And one of the things that you notice is that the TATA-Binding Protein favors the bending of that DNA molecule. The bending of the DNA molecule turns out to be important in opening up that complex, which happens further along the process as other proteins bind. So the bending is important. And it's not uncommon that we see bending associated with DNA binding proteins. Does anybody here remember the last time we talked about bending with a DNA binding protein? What was it? Not the, quite, you're close though. Restriction enzymes. Restriction enzymes did that, right? They bent that, and that bending facilitated the breaking of the bonds. In this case, it's bending it and facilitating the opening up of the strands. Okay. We talked about heat shock, and I talked about heat shock with respect to bacteria. And I told you at the time that heat shock genes are present in almost all cells, and this shows an arrangement of sequence elements, promoter sequence elements for the heat shock gene of of eukaryotic cells. I believe this one's actually from humans. And what we see is here is an element. Here is a conserved sequence, and it's present not once, but it's actually present 4 times. This is not at all uncommon for sequence elements that play roles in transcription in eukaryotic cells. In many cases, they will be present in multiple forms, and what those multiple forms allow, or not multiple forms, but multiple numbers, those multiple numbers allow ahead of a transcriptional start site is more opportunities for the proteins, the transcription factors that bind those, to bind and get transcription started. So, not surprisingly, we can actually make this actually be stronger, more transcription occurring if we have more elements ahead of the gene. Okay, so I've talked about enhancers. I want to just say a brief word about ribosomal RNAs in eukaryotes. You remember in eukaryotes that we have 3 different RNA polymerases. The one I've been talking about so far has been RNA Polymerase II, which makes the messenger RNAs. RNA Polymerase I makes the ribosomal RNAs. And look at what we see here. We see that kind of like we saw in prokaryotes. In prokaryotes, we saw the ribosomal RNAs and the tRNAs that were synthesized in one long piece, and then it got chopped up into individual components that gave the cell the ribosomal RNAs and the tRNAs that it needed. We can see here in the case of the ribosomal RNAs in the eukaryotic cells that 3 of the ribosomal RNAs, they're called 18S, 5.8S, and 20S, are made on a single pre-ribosomal RNA. That's what this is called here. It's a long, big piece is basically what it means. And in order to get the finished ribosomal RNAs, 2 things have to happen. One thing that has to happen is these intervening sequences have to get chopped out, and there are nucleases that will do that very nicely. The second thing that has to happen is that there's chemical modification that's occurring. I talked about chemical modification with respect to RNAs in prokaryotes. We also see it happening in eukaryotes, and we see modification of individual bases that are happening on these ribosomal RNAs. So at the end of that, we have ribosomal RNAs that are ready to function, and I'll talk more about those ribosomal RNAs later today. Okay, and the last RNA polymerase I'll talk about is RNA Polymerase III. RNA Polymerase III makes all of the transfer RNAs, and it also makes the very shortest ribosomal RNA. It's known as the 5S ribosomal RNA. Like we saw in prokaryotic cells, the transfer RNAs are in fact made as part of a larger transcript. That's not shown here. And there's some processing that has to happen, some modifications that have to occur to make the final transfer RNA. Here is a sequence called a leader sequence that has to be chopped off in order to make this final transfer RNA. So you see the chopping off of that. And like we saw in prokaryotes, we cut off a little piece at the 3' end and we add that CCA at the end. So the CCA is a good marker for the 3' end of the transfer RNA. You'll also notice that there are structural features in this transfer RNA. I'm not going to talk about them here, but I will talk about them in just a bit when I talk about translation. Okay, eukaryotic messenger RNAs, I noted earlier, are quite a bit different from prokaryotic messenger RNAs in that there's a considerable amount of modification that happens to these eukaryotic messenger RNAs. Okay? One of these is that the 5' end of eukaryotic messenger RNAs is chemically altered. And it's altered in an unusual way. It's altered by putting something on it that we refer to as a cap. Okay? A cap is a nucleotide. It's actually a modified guanine nucleotide that is put onto the 5' end of the messenger RNA in sort of a backwards fashion. What do I mean by that? Well, if we look at how the messenger RNA goes, just like DNA goes, we see 5', 3', 5', 3', 5', 3'. So the 5', the 5' end of one is attached to the 3' end of the previous one. All right? When we look at the very 5' end of a messenger RNA in eukaryotes, we discover that this modified guanine nucleotide has been attached not in a 5', 3' orientation, but in a 5', 5' orientation. Now that seems a little odd. Is it just random that they do this? And the answer is no, it's not random that they do this. A couple things happen. One is the 5' end now is clearly marked as being the 5' end. And though the cell could probably figure that out, that does give some identity. It turns out that this unusual structure is actually used in translation of eukaryotes. So this provides us sort of a handle. This is very good. The more important reason that there's a cap on there, however, appears to be that the cap gives stability to the messenger RNA. What do I mean by stability? All right. Well, let's compare eukaryotes and prokaryotes, and I think you'll see what I'm talking about. A prokaryotic cell, let's say an E. coli cell, has a complete life cycle of 25 minutes when it's got food. 25 minutes. It starts from scratch, it goes all the way through its life cycle to division, and starts over in 25 minutes. Okay? If we think about the varying needs of genes that prokaryotic cells have as a function of where they are in their life cycle, 25 minutes is not much time to change. I need a DNA polymerase now, but I don't need a DNA polymerase later. I need ribosomes now, I don't need ribosomes later. Okay? One of the ways that cells control how much protein they have is whether or not they transcribe. But once an RNA has been made, the cell needs to be able to control that as well, because that RNA is sitting there, and if the cell continues to translate something it doesn't need, it's wasting energy. So cells, in addition to controlling when RNAs are made, also controls how rapidly it degrades them. The half-life, meaning the time it takes to destroy about half of the messenger RNAs, the half-life of a messenger RNA in a prokaryotic cell is on the order of a couple of minutes. It gets used for a couple of minutes and it gets broken down. Because if it sits around too long and the cell starts using it when it shouldn't be using it, it's wasting energy. In a eukaryotic cell, the division time is about, depending on the cell, 24 hours on average. A much longer cycle. The lifetime, that is the half-life of a messenger RNA in the eukaryotic cell, is on the order of hours. Much longer it's around. One of the things that helps it to be around longer is the fact that its 5' end is protected from degradation. When we look at how the RNAs in the cell are degraded, they're degraded in two ways. They're degraded starting at the 5' end and chewing inwards, or else they're degraded at the 3' end and chewing inwards as well. That happens in prokaryotic cells. In eukaryotic cells, that cap stops that 5' degradation. So the cap is protecting the 5' end from being degraded by nucleases. Very important feature of the cap. Okay. Another interesting modification that happens to eukaryotic messenger RNAs occurs at the 3' end. At the 3' end of eukaryotic messenger RNAs, but not prokaryotic, okay, only in eukaryotic, we see what's called addition of a poly-A tail. The poly-A tail, as its name suggests, is the addition of a bunch of A's to the end. Now this figure shows a little bit about how that process occurs. Here's the messenger RNA being made. The cap has already been put on. The cap gets put on pretty quickly, pretty early. It's gone along, and the polymerase has finished synthesizing this sequence AAU-AAA. This AAU-AAA is a signal to the proteins that put the poly-A on that, hey, we're about ready to put the poly-A on. Okay? So after the polymerase has gotten a little further down, this system, this specific endonuclease, will say, okay, I'm going to chop a few bases off. I'm going to chop off about right here, for example, and then a polymerase is going to come along and put hundreds of A's on there. Now, there's a couple useful features of this. One is once this sequence has appeared, the RNA gets chopped. Termination isn't as much of an issue here, because the chopping provides the defined 3' end. The other interesting thing is actually the addition of the poly-A's. If I put a few hundred poly-A's on here, and what I just told you was that the stability of messenger RNAs is determined by whether they get degraded at the 5' end or at the 3' end, we start thinking that, well, the longer the poly-A's, the more poly-A's there are on there, the further a nuclease has to chew in before it gets to the meat, that is the coding region. The length of the poly-A tail helps play a role in determining how long a given messenger RNA exists in a eukaryotic cell. The longer the poly-A, the longer it'll be around. The shorter the poly-A, the less time it'll be around. We don't see either of these modifications, either the cap or the poly-A, in prokaryotic cells. Okay. Now, there's one other class of RNAs that I want to talk about briefly, and I'll say a word about them also when I go to talk about gene expression. And these are relatively recently characterized RNAs called microRNAs. They're typically made by RNA Polymerase II or III. III, recall, is involved in making small RNAs, and microRNAs get their name from the fact that they are indeed small RNAs. These RNAs play roles in helping to control gene expression. We'll talk about that later, but it's a very interesting phenomenon by which they do this. And their discovery is relatively recent, 1990s. Okay? When people started working with RNAs, they discovered there were zillions of little tiny RNAs that were in the cell. And when they first started characterizing these little tiny RNAs, they said, oh, it's just pieces of bigger RNAs that have been broken off. It's all this junk. The cell, of course, is chewing up its RNAs, so it's not surprising we'll have all these little pieces here. And they largely didn't pay attention to them. Well, of course, not paying attention to things in biology is a mistake, because as researchers subsequently discovered, those little RNAs that were there had a very, very important role, part of which you see here. Okay? What this scheme is showing us is the synthesis of these small RNAs, microRNAs, and the processing of them. So here is a small RNA that's been synthesized by an RNA polymerase. You'll notice it has, hey, there's a stem loop. You've seen that before. It has a stem loop, but this stem loop is doing something different than what you saw before. All right? It's not acting as a jack to lift things up. Instead, what this guy is doing is providing a structure that's recognized by some proteins that do this processing that you see here. Okay. What does that mean? Well, this final processed RNA is used by a system in the cell, this is a protein called argonaute, to decide whether or not to stop translating the messenger RNA that's complementary to this. So this is providing, and as I said, we'll talk more about this later, but this is providing a way for cells to regulate gene expression in yet another way. After the messenger RNA has been made, there's other ways of controlling how much of a protein is there, and this microRNA plays a role in that process. As I said, we'll say more about it later. Okay, questions about what I've been saying here? Everybody's ready for the weekend. Me too. Yes, sir.
I'm sorry, could you repeat just a little bit of the last part of the microRNA?
Okay. So with respect to the microRNA, the microRNA system provides a way, another way of controlling how much of a given protein is present, and this plays a role, as we shall see, in regulating the translation of sequences complementary to this. Okay? Sequences complementary to this. Yes? Okay, which RNA polymerases make these microRNAs? RNA polymerase II and III. And these guys are fairly short, okay? This final product over here is about 25 nucleotides long. So this guy over here, as it's made, is 50 or less. It's not very big. Okay. Well, there's other modifications that happen to RNAs in cells, in eukaryotic cells. And I want to talk about one interesting one here. There's a process called editing. And editing of RNA occurs in, I'm not totally familiar, not totally happy with this term editing, because as you will see, editing can occur in a couple of ways. This editing that you see here is a chemical editing. And it's not editing in the sense of proofreading, which is what we thought of with respect to the same term in DNA replication. But the editing is in terms of modification. So perhaps a better description would be this is a chemical alteration of the RNA. What this shows right here is a messenger RNA that codes for a protein. It turns out it codes for two different proteins, the same messenger RNA. And which protein is made depends upon which chemical modification occurs to the messenger RNA. Let me explain. One protein that can be made is called ApoB100. ApoB100 is a protein that's made in cells that make chylomicrons, intestinal cells. Okay? So ApoB100 is an important protein in chylomicrons. A related protein is made by liver cells. It's called ApoB48, and it's made in liver cells. It becomes a part of LDLs. Now, as you will see as you look at this, there's a part of the protein that's common in the two. The blue part of the ApoB100 is the blue part of the ApoB48, meaning that the first half or so of the protein is identical. Well, what's different between the two of them? Well, it turns out that in about the middle of that messenger RNA, there is a sequence CAA. In intestinal cells, that CAA is translated without any change. And that allows the synthesis of this large protein up here. On the other hand, in liver cells, they have an enzyme that converts the C to a U. It involves removal of an amine group. Well, a UAA is a stop codon. It stops translation. So in liver cells, this entire message doesn't get made. It gets truncated because the C is converted to a U, and you make this shorter protein. Now, this is called RNA editing. It's actually chemically altering at least one of the bases of the messenger RNA. Now, there's another place where the term editing is used, and I'll mention it briefly, but you're not responsible for it. And that is that there are a group of lower eukaryotic organisms called trypanosomes. They have a very bizarre type of editing that they do. At many places within a final messenger RNA in trypanosomes, they will physically insert A residues. So you might have a sequence CGG, and this would come along and make it a CAGG or CAGAG, right? And this editing is bizarre, I'll be honest with you, but it's necessary for the proper translation, the proper synthesis of protein. The term RNA editing is also used to describe that process. So that's why I'm not totally fond of using this term editing. Okay. The last thing I want to talk about with respect to alteration of RNAs is something else that's pretty much unique to eukaryotes. There's only one or two very minor examples in prokaryotes. So for our purposes, we're going to say that splicing is a phenomenon that only occurs in eukaryotes. Splicing is an alteration of a messenger or of an RNA. It doesn't have to be a messenger RNA. It could be a ribosomal RNA. It could be a transfer RNA. It could be a messenger RNA. Splicing is a covalent alteration of the RNA after it's been made. And that covalent alteration that we see removes a segment, at least one segment, from that finished RNA. The removed segment has a name. It's called an intron. So an intron is a segment of RNA that is excised from the RNA after it's been made. Isn't it? All right? The segments that remain are called exons. So we see here's an exon, and in the process of this splicing phenomenon, this intron will get removed, and the AG will get joined over here to this G over here. Okay? So splicing is removing this central piece and joining the two other pieces that remain together. Okay. Well, I'm not going to go through all the details of splicing, but I do want to talk about the mechanism in very brief terms. Splicing allows cells to put together coding pieces, if it's a messenger RNA, allows cells to put together coding pieces in very flexible ways. It allows for tremendous diversity of proteins using a given sequence, and I'll show you examples of that in just a bit. To understand the splicing mechanism, we have to look at, and this is not the best figure, but we have to look at the precursor RNA. This is what it looks like here. When cells splice, they have to recognize the place where the sequence is to be removed from. It's shown here, starting in pink. The other end, going over here, and then that actual piece has to get removed. Well, it turns out that when we examine the pieces that get removed, that is the introns, there is a little bit of sequence information that tells the cell, hey, this is an intron that's got to be removed. Here's a sequence that's common near the 5' end of the splice site. That is the place, the 5' end of the fragment that gets removed. You see this sequence here. The only part of the sequence that is found everywhere is the GU. GU is always found at the 5' end of a splice junction. At the 3' end, that is the other end, the only thing that's always found is that those terminal bases that you see, AG. Okay? The AG is found everywhere. There's one other thing that is commonly found within the intron, and it's this little sequence right here. This is a sequence. The Y stands for pyrimidine. It's a pyrimidine-rich sequence, and that pyrimidine-rich sequence is usually found about 20 or 30 base pairs away from the 3' end of the intron. So 3 conserved things that we see. A 5' GU, a 3' AG, and a pyrimidine-rich region about 20 or 30 base pairs away from the AG. Introns can be enormous in size. I worked with one time that was about 10,000 base pairs long. They can be short. They might be as short as 50 nucleotides in size. Okay? Now, the mechanism that's involved in this is, first of all, there's a nucleophilic attack. And the nucleophilic attack occurs by a hydroxyl that is always on the A in the middle of that pyrimidine-rich region. The nucleophilic attack is on the phosphate at that junction. That attack results in the formation of a covalent bond between the 5' end and the 2' hydroxyl on that RNA. Now, what we've made in the process of making this covalent bond is something that scientists call a lariat. This looks like a lariat. And we're going to see the lariat finished off in just a second. Okay? Here we are. The lariat isn't quite done because we haven't finished off the 3' end. Now, to make the 3', to cut the 3' end off, this AG, you see the 3' hydroxyl of this G right here, attacks the phosphate at this junction. That results in the joining of this to this. So we see there's 5' AG, and then there's that G that's right there. So we've joined this piece here to this piece here. In the process, we have cut out a lariat. What good is the lariat? Everybody always asks me. Okay. Typically, lariats are simply degraded. But a few lariats have coding in here. And occasionally, there are genes that are actually translated inside from the lariat. It doesn't always happen, but there are a few cases where that does happen. Lariats are just mostly thrown away. Okay? Now, why do cells do this? Well, like I said, it allows cells to mix and match coding regions. If we're talking about a protein, this mixing and matching can give us quite a variety of proteins. Let me show you an example here. Okay? If we look at different splicing, wrong one. Here depicts a pre-messenger RNA. That's an unspliced messenger RNA for a protein known as calcitonin. In neuronal cells, 1, 2, 3, pink, 1, 2, 3, pink, one version of calcitonin is made. In thyroid cells, a different version. Same coding region, but now the blues get attached to that. This mixing and matching of exons provides diversity. It provides for different proteins to be made in different tissues, starting with the same coding region. And when we talk later about the immune system, one of the things that's needed for a robust immune system is diversity of the proteins and the antibodies. And this provides tremendous shuffling. So splicing turns out to be very, very important for giving diverse proteins for cells. Okay. Now, let's see. There's something, a term that you should be familiar with, and the term you should be familiar with is a spliceosome. S-P-L-I-C-E-O-S-O-M-E. Spliceosomes are complexes that perform the splicing. The complexes, as you can see on this screen, include proteins that you see labeled U1, U2, U3, etc. And those proteins have a name. They're called SNRNPs, small ribonucleoparticles. SNRNPs is probably the way you'll know them. Okay? These proteins play important roles in the splicing process, and as you will see, the proteins carry with them, in many cases, an RNA of their own. So the spliceosome complex that comes together to do the splicing contains protein and RNAs. That's not very different from what we see happening in translation, because the ribosome, which has a very similar name, is comprised of proteins and ribosomal RNAs. Now, I'm not going to go into the details by which this happens. You'll thank me for that, I think. Okay? Or maybe you won't, I don't know. But suffice it to say that the proteins and the RNAs that are here allow for the alignment of these sequences to occur that need to happen. Okay? So let me show you that briefly. That's not it. Dang it. That's not it. Dang it. That's what I'm after. Okay. Now, this is up close and personal with a pre-RNA that is about to be spliced. As you can imagine, we want to arrange things so that the pieces come together nicely for the splicing to occur. What you see here are some of the RNAs that those snRNPs are carrying. And these RNAs, not surprisingly, are complementary to sequences inside of that intron. So the snRNAs are complementary to sequences inside the intron. They, combined with the snRNPs, give a geometrical arrangement of the exons so that they can come together and be joined together properly. They don't go flying off somewhere. Okay. That's what I think I want to say there about those guys. No, actually, I don't want to say that. What was I going to say? Various functions of the individual ones. I'm not going to hold you responsible for that. Okay. All right. So splicing, I've given you one of the very good sides of splicing, is that you get a lot of diversity. And a lot of diversity allows a cell to make many proteins from a given sequence, and in eukaryotic cells that turns out to be very useful. Like any process, if there are problems with the process, we can imagine there will be some downsides, and there are downsides. It's possible in some cases to have a single base pair mutation that will change the splicing. If splicing is critical for mixing and matching the exons, we could imagine that if we change the sequences that tell the cell whether to splice or not, we might have some problems, and that's exactly what we see. Okay? Thalassemias are a really good example. Thalassemias are mutations that affect the ability to make globin genes for making hemoglobin. Some thalassemias arise from having a mutation that changes the splicing tendency. Changes the splicing tendency. Well, in this case, we see a sequence that is where an A is converted to a G. That creates a new 5' splice site. And the result of that is that a stop codon that otherwise would not play a role now plays a role in stopping translation. Okay? Normally, this wouldn't even appear in a finished product because this entire intron would get spliced out. But if the intron is spliced here instead of being spliced here, now we can make a globin that has, a stop codon where there shouldn't be one, as well as some other sequences. That's going to create a globin that's got major problems. And globins are obviously important for making hemoglobin. So this single base mutation caused a severe problem to a person who suffered this mutation. Okay. There are other diseases where this is known, and again, I'm not going to go through these, but I just show these to you to indicate that, again, splicing can have problems arising when mutation gives rise to things that alter splicing sites. Okay. Now, that, I think, is what I want to finish up with there. Are there any questions about that? Everybody's tired. Everybody's ready for the weekend. And is he going to start translation or not? Yeah, he's going to start translation. Oh, 5 more minutes till the weekend, guys. Yes? I'm sorry? Yeah, good question. Does splicing occur before capping and so forth? Capping is one of the very first things that happens. But splicing can happen before the RNA is complete. Okay? Actually, there's a figure I wasn't going to show you, but I'll just briefly show you since you've asked. Splicing can happen before the synthesis of the RNA is complete, and that actually is shown right here. That carboxy terminal domain that got phosphorylated provides attachment points for capping enzymes, for splicing enzymes, and for polyadenylation. So we can actually see capping occurring, we can see splicing occurring, and finally we can see polyadenylation occurring because of these things that are being carried on the end of the RNA polymerase. So yes. Now remember, we don't see translation happening because in a eukaryotic cell, we're in the nucleus, and the translation is done out in the cytoplasm. So all this processing has to get done before the messenger RNA makes it out to the cytoplasm. Okay. Now I want to emphasize that splicing is not solely happening to messenger RNAs. Ribosomal RNAs and tRNAs can both also be spliced. Okay. Brief introduction to translation. Translation in cells is obviously critical because cells have to make protein. Translation is the process of converting messenger RNA sequence to amino acid sequence. It's the process of making a protein. Proteins, you recall, consist of amino acids linked together by covalent bonds called peptide bonds. And a ribosome is the protein ribosomal RNA complex that does the translation. It grabs a messenger RNA, it finds the right place to start the synthesis of the protein, and it slides down that messenger RNA. You can see this guy is sliding down the messenger RNA. There's the messenger RNA right there. And in doing so, in sliding down, it's covalently linking together amino acids, and those amino acids are squirting out the top as a polypeptide. You'll notice EPA. We'll talk more about those because these are specific regions within the ribosome that have functions. Okay. This shows the formation of peptide bonds. We see that one transfer RNA that carries an amino acid is paired up with another transfer RNA carrying an amino acid. The amino acid is transferred from one to the other, and a peptide bond is formed. We have just formed a dipeptide, 2 amino acids covalently linked together. Okay. Translation is, the accuracy of translation is important, but also important is getting the protein made. We talked about how in RNA, when cells made RNA, that fidelity was important, that is, few mutations, but I said that it wasn't as important as DNA was. Keeping mutations out of DNA was important because cells passed that DNA down from one generation to the other. They didn't pass their RNAs down from one generation to the other. And similarly, they do not pass down their proteins from one generation to another, at least not for any long period of time. The fidelity of the translation is on the order of 10 to the minus 4th to 10 to the minus 5th. And we can see that that means that for a protein that's about 1,000 nucleotides long, that at least 90 to 99% of those will be perfect in sequence. Why don't we want it to be 100%? Well, we could. But it would take so long to proof it that it would take us a long time to make a protein. And cells are more interested in cranking out protein. So they sacrifice a little bit of fidelity to make translation move more rapidly. Okay? Now, I'll stop there. How's that?